Okay, good evening, good morning, good afternoon, everybody. This is SBOAJC Alumni Batch of 2003 Learning and Unlearning Initiative for Better Living and Better Understanding the World. We are in season 2, session 6. Overall, we are at the 12th session. Today's topic is Fish and Art by Jeevitha. Jeevitha is doing a repeat. She did a first session in Basics of Genetics, Genetics 101. I think it was session 2 or session 3 in season 1. She's back here. I think this is also related to genetics, but somewhat deep and somewhat more contextual in a societal problem that we face in some sense. So over to Jeevitha. Thank you.
Okay. Hi, friends. Good evening, good morning to our applicable. Today's topic, fish and art. Am I audible?
Yes, loud
Okay.
and clear. Yeah.
Okay, fish. It's not the fish we fry and eat, but it's fluorescent in situ hybridization. It's a molecular technique which is used to detect your DNA sequence using a labeled probe, which is fluorescent labeled probes. Okay, this is the principle. Your For a target DNA, you design a fluorescent probe and bind it to the target, and then it's viewed through the fluorescent microscope, which shows your presence or absence and the location of the gene in a chromosome through the microscope, with which you can detect the abnormalities or possibilities of whatever we can do with that.
Jeevitha, can you go back? Sorry, this is too technical in some senses, so I might ask questions if in case.
Okay, no
Yeah,
issues.
can you go back? Can you go back?
Yeah.
Molecular cytogenic technique. Okay, good. Because the reason that I'm asking this question is, uh, I attended some other event yesterday where we were exploring or trying to understand molecular diagnosis itself as a company or a topic. Okay, so
Okay.
from a layman standpoint, I would request you to explain, say for example, these probes and whatever these technical terms in a much more lighter manner, so, from a molecular diagnosis standpoint, for a layman, my understanding is we give our blood samples. Okay, it can be
Okay.
care, it can be any medical or something, but
Yeah.
they are not necessarily doing the end this thing. So, depending upon the complexity of the diagnosis, they send our samples to some other party who are specialists.
True. True.
Those specialist parties are only coming under this molecular technique or any molecular technique for that matter, right?
Yeah.
Yeah, understood. Okay, okay,
True.
okay.
This is done in even for you use this technique in even egg or sperm. We can do it even with egg or sperm.
Understood. Okay.
Okay, our DNA is basically composed of four components, right? I mean, the four sugars: adenine, guanine, cytosine, and thymidine. The four phosphate groups.
Yeah. Okay.
So now, when you have to prepare a probe, your target DNA will be within this.
What do you mean by probe? Can you define a probe here?
See, if for example, if your target is for AT G, so adenine, guanine, thymine, cytosine, yeah, GC, ATGC, it make a keep 2 or 3 repeats of it. If there is a problem, instead of T, you get another G, AGG. That actually is a issue there.
In terms
And
of
after
sequencing, in terms of sequencing. Okay.
sequencing.
Okay.
Okay. So that error might even create a trouble during the what to say error and give you some issues.
Is this as same as genome sequencing? Because I did genome sequencing for
Yeah,
some
yeah,
years.
yeah. It's
Yeah,
like a
equal to
protein
that.
sequencing.
Protein
Protein
It's like a
sequencing.
sequencing.
genome protein
Okay.
sequencing.
Mine
It's
was, my
similar to that.
report was like, if I remember, I don't know, 183B D2 I don't know, but in particular, because my I will share my own experience later. But mine was a chromosome defect, and that was reflecting when they did genome sequencing. And after my daughter was born, just to be doubly sure that there is no abnormality in her, we did a genome sequencing to her, and then the same thing across all things. If in case you imagine this a 10-meter rope, and then the fifth meter, fifth feet, second centimeter is what if in case everything is blue and that was yellow for me, for my daughter also it was yellow, right? So doctor was, if this yellow is not happening to our daughter only is a concern. If it is replicating, it's not a concern because you don't have a concern, right? So okay, understood. Okay, so genome
So,
sequencing is the same as DNA sequencing. Is it right way?
sure.
Okay, okay.
So to target that, because it might to target that, you prepare a specific probe which goes and actually fuses with the target DNA. Almish, are you clear? Target DNA, and that shows the absence or presence of the gene location in a chromosome. Okay. Okay, the types: centromeric fish. It detects the whole chromosome with the centromere as a target. Locus specific, that detects a specific gene on a particular chromosome, detects a specific gene. And whole chromosome painting, it gives a color to the entire chromosome. And multicolor fish identifies multiple chromosomes at once. If you have the same gene in 2 or 3 different chromosomes, then it identifies all those. Okay. Okay. Okay, the protocol for fish sample prep. Cells, that is, the chromosomes are in the metaphase stage or an interphase nuclei. They are fixed on a slide. And next, the process is denaturation, where the target DNA and the probe DNA are mixed together and heated to make them single-stranded, so that the probe DNA can go and attach, which is the process of hybridization. Once the probe DNA and target DNA are attached, then we watch the excess unbound probes using a buffer, and then the slide is viewed under the microscope to check for signals so that they can do the diagnosis for the case.
But in what context for whom facing what is this fish required in the first place?
I'm coming to that. The
Okay.
next is that.
Oh, okay.
Okay, this is the picture, the proper picture. See, you have a slide which is what fixed with a sample. Here you have the target DNA, the probe DNA, which is fused using which is fused and hybridized, and this can be viewed under the microscope. And when you have when the target is achieved, then you have the fluorescent visualization as shown here. Okay, application for fish.
Can you go back a little? Can you go back?
Yeah.
So here you're saying this is a DNA because its helix is DNA.
Yeah.
Okay, and inside that whatever that green yellow that band இருக்கிறது வந்து
ஆமா அது
அது
fluorescent probe.
fluorescent problem, is it an artificial substance or something? What it is?
See, you know through genome sequencing, you know the sequence of the probe you have to target.
Okay, probe
Sequence.
na the section in which you need to analyze. Okay.
The target is a section in which you need to analyze the defect.
Okay.
So based on that section, you actually prepare a probe, which is mostly done by the pharmaceutical or genetic companies. Okay. So we purchase based on that, and then we do the protocol as it is.
Understood. Okay.
No,
And
can I try to explain it for Madanish here? Like,
yes,
so,
yes, you
Yes,
can.
please.
Please. As much
basically,
as plain English is possible, please.
see, DNA is a double helical structure, right?
Yes,
But you
yes.
also know that if it is an A, it will only, connect with only T, right? Correct me if I'm wrong with the protein sequence. If it is A, adenine, it will usually only connect with thymine or something. There is a pair.
ATGC, yeah.
Correct.
There is a pair.
Some sort of
So
logical
what they,
sequencing. Okay.
yeah. So what they do is, let's say I want to target a sequence A, G, T, C, O, A, G, C, something like that. Like let's say, like a long sequence. What they will do is they know this is the sequence they want to target. So they create an opposite pair of it. If it is A, it means T. G means C. Like that, they create another pair, but they'll also add that fluorescence to it. Fluorescence will be some kind of bonding, some kind of oxygen bonding or something will be there, and will create fluorescence. And then
See,
when
for
they send it
C2,
near that particular DNA sequence, it will remove the old bond and create the new bond with this fluorescence. Now, when you see through microscope, you'll see the fluorescence now, whether the sequence is correct or wrong or like whatever it is. So that's
Okay,
how it will.
then is this almost equivalent to what I am reading or what I've read as gene editing? No.
No,
no.
that's different. That's CRISPR-Cas9. They use adenosine targets, and then they'll clip the.
This is not
That's
gene editing. So it's
it's
DNA,
like
na?
fish.
Oh, okay.
This
Understood.
this was the precursor to that. Based on this technique only, they created the CRISPR-Cas9 editing.
Understood. And that's more deeper. Gene editing is much more deeper.
It's much more deeper.
Okay. Okay. Understood. Okay.
Okay. Application for fish. You detect chromosomal abnormalities. It's used in prenatal diagnosis and cancer diagnosis and gene amplification of HER2 in breast cancers. HER2 is a very big expansion. If you want the expansion, I can give you that. One sec. Okay, I'll message you guys about the HER2 expansion. It's used in infertility and reproductive medicine and identification of microdeletions. When you do a normal karyotyping, that is which is using done using your blood samples, it you can't see microdeletions and all that. You can just see the presence or absence of a chromosome or a multiplication of chromosome. So in this protocol, you can even check on microdeletions.
Understood. So just sharing my experience because just to combine everything together instead of keeping it last.
Yeah.
So I had an issue, or rather, it took time for us to conceive, etc. That's a separate story. I can share a separate session only. But if I remember in 2019, first was karyotyping for me.
Yeah.
And then specifically the prescription was
Mm.
Y chromosome microdeletion. Check for Y chromosome microdeletion.
Uh,
So what
you
you're
will check
saying
all
is,
that. Mm.
what you're saying is normal karyotypingல அந்த Y chromosome microdeletion இருக்கா இல்லையாங்கிறது தெரியாது.
தெரியாதாமா.
அப்போ நான் காசு இப்போ நான் 20,000 or something நான் ஒரு பில் தான் எனக்கு கொடுத்துருப்பாங்க. அதுக்கு உள்ள Y chromosome microdeletionக்கு fish யூஸ் பண்ணி they would have given me the report.
Exactly.
Understood. Okay. Okay. Okay.
See, the HER2 expansion in a human epidermal growth factor receptor.
Growth factor, yeah.
Okay. Hi, Abhi.
Hi.
Okay, advantages of fish. Highly specific and very sensitive. Can be used on non-dividing and interface cells. Faster than the traditional karyotyping. Because traditional karyotyping takes more than 2 weeks time. Whereas this is immediate. They will do it in a few days. Limitations. Detects only the target DNA. Requires prior knowledge of DNA sequence. And it's pretty more expensive than the staining technique. Than your traditional karyotyping. This is more expensive.
I think Sneha has a question.
Yeah.
Thanks. Thanks. Yeah. I don't know if you explained what is karyotyping?
That was in the previous presentation, Sneha.
Okay. I'll
நான்
Google it. Okay,
ஒரு 2
no
mark question
problem.
ஆன்சர் பண்ணுமா ஒரு 2 mark question.
See, karyotyping is you actually you have 23 pairs of chromosomes. Okay, that you actually do a blood sample, draw a sample from your patient, process the sample, and view the sample through the microscope. After staining them, there you can check the chromosomes from 1 to 23
For
pairs.
in the lens of abnormalities. Now, I'll give you an example. My
Abnormalities.
case was the issue that I had, or it was a function of something retrospectively, whatever, logical analysis. In my karyotyping, as he told, that's a report of all 23 chromosomes. Okay, which means அந்த ஆக்சுவலா சூப்பரா பார்க்கணும்னா நீ மானிட்டர்லாம் பார்க்கணும், லேப்ல கலர் கலரா தெரியும். உனக்கு கொடுக்கறது வந்து டிபெண்டிங் அமான் த
Black and
லேப்
white தான்
லிஸ்டிங்.
இருக்கும்.
ஜெராக்ஸ் போட்டா ஒண்ணுமே தெரியாது. ஜெராக்ஸ்லயும் அந்த ஷேட்ஸ் வச்சு
You
பிளஸ்
can't
அந்த
identify
அந்த
anything.
அந்த
Yeah.
பிரிஸ்கிரிப்ஷன்ஸ் அண்ட் சம்மரியை வச்சு ஆப்வியஸ்லி எக்ஸ்பர்ட்ஸ் ஆர் எக்ஸ்பர்ட்ஸ், தே வில் ரீட். பட் த பெஸ்ட் வே டு ஐடென்டிஃபை திஸ் இஸ் த கலர் பிரிண்ட் அவுட் ஆர் த லேப் திஸ் திங். So each chromosome there will be actual pictures. In my case, in my fifth chromosome, which is like this X, assume X, right? That is one. If we would have studied, if you remember or if you don't remember, I'll remind you. There is P band and Q band or something, right? Which is up and down. So
PM and QM, yeah.
in my fifth chromosome, inside the P band, there was one small inversion. Inversion-னா இதோ இது இப்படி
கீழே
இருக்கிறது, இது
இருக்கிறது.
இப்படி இருக்கிறது பதிலா இது இப்படி இருக்கு, அவ்வளவுதான். Right? In terms of whatever that I don't know the technicality, but that had repercussions on the issue, or rather that was a logically connecting the dots. That was one reason that I had the issue, which was solved through IVF cycles or ICSI cycles, etc., which is second part of the presentation today. I will also share my experience there. But இந்த ஃபிஃப்த் குரோமோசோம்ல பி பேண்ட்ல இந்த இதுல இருந்து இன்வர்ஷன் இருந்ததுன்னா இதுக்கு பாசிபிள் கேசஸ் ரிசர்ச், ரைட்? இதை நீ போயிட்டு இந்த இன்வர்ஷனை வந்து நான் இப்படி இருக்கிறத வந்து ஐ கேனாட் கோ அண்ட் ஆப்ரேட் அண்ட் தென் டூ லைக் திஸ். இட் இஸ் வாட் இட் இஸ், அவ்வளவுதான். அது ஃபிஃப்த்துக்கு பதில் செவன்த்ல ஒரு பேண்ட்ல இப்படி இருந்ததுன்னா, எயித்துல இப்படி இருந்ததுன்னா, எவ்ரி திங் ஹாஸ் இஸ் ஓன் பிரான்ச்சஸ் ஆஃப் again, right? Abnormalities is not an abnormality for me, right? It is not, fortunately it is not related to my senses, it is not related to my motor thing, everything, right? But ஒரு குரோமோசோம்ல ஒரு abnormality இருக்கிறது வந்து, abnormality can be real abnormality affecting you physically or mentally.
And mentally, yeah.
It need not, for me it's not a case, right? But இது வந்து அந்த issue அந்த situation வந்ததுனால we were subscribed to do it. For normal people who are not facing any challenges, stereotyping, it's a very big word. It's a very technical word, right? Nobody
They won't even hear about it.
Exactly.
Oh, but thank you so much for explaining both. Thanks a lot. And I'll also look up your earlier presentation. I know there is a link to it. So thank you both.
Yeah, thank you. Okay. Now, role of fish in infertility and reproductive medicine. It's used widely to detect your chromosomal abnormalities, which causes infertility and repeated pregnancy loss and implantation failure. An evaluation of male fertility performed on sperm cells to detect abnormal chromosome number or sex chromosome abnormality, as you said, for Y chromosome. And it's commonly used to analyze chromosome abnormalities in X, Y, 13, 18, and 21. Okay. Any doubts? People, any doubts?
How I'm assuming this has an equivalent to the female part as well.
Yeah. That is.
But second part I understand because I have experienced that. But you are saying performed on sperm cells to detect abnormal chromosomes.
See,
how do you is இப்போ நம்ம blood sample எடுக்கும் போது அதுல இருந்து செல் வந்து there are again
fish is not only done on blood samples, okay? It's done even on your egg and sperm.
Understood.
Ovum and the sperm is also used in this because
it can be used, is your point?
No, it is actually. That's how you filter when, see, if you're going through artificial reproductive process.
Correct.
If you have a mosaicism, meaning you have half of your normal the cells have normal chromosomes and half have defective ones. So your egg and sperm, egg and sperm will also have the same issue.
Understood.
So to filter that.
Okay. Because maybe relating to my course of actions, we had a ICSI cycle. So I think what you are saying is when they chose the embryo, right? Or even before they chose, even before they formed the embryo, what sperms and what eggs to choose.
Exactly. They
is
check
where this comes. Okay, understood.
Yes.
Understood. Okay.
So this you don't have to process through what to say centrifuging or whatever and break the sperm or egg.
And
You just
yes,
have to
without that you
target.
can do.
You
Understood.
can do. So this is not, you don't have to retrieve more samples of egg
Understood.
or sperm.
And it also can be used, as you said, it can be at a prenatal stage, because once an embryo is formed, depending upon the complexity,
Exactly.
a small tissue from that embryo itself can be sent to this. But
Send
we
to
did
this,
not do
yes.
this for our daughter or son
Exactly.
because there was no need. It was more a comfort level. Na? So, okay,
For
understood.
your satisfaction, you did it so
Yes,
that you can be happy. That's it.
understood. Okay.
Okay. Evaluation of female fertility. Detect structural chromosome abnormalities in oocytes and somatic cells. Somatic cells is your normal blood cells. Look, white blood cells. Only your white blood cells have the nucleus, okay? And identifies Turner syndrome mosaics. As I said, your mosaics are half and half, 45X and 46XX. Balanced translocations, meaning part of chromosome from one, part of a chromosome can move to the other chromosome and settle there. You won't know without the fluorescence in situ FISH technique. Some experts can even do through the traditional karyotyping process, because when they keep viewing case samples, they'll know the length and the size of the chromosomes. We actually identify the chromosomes and number them accordingly when we have to check through slides. So when, say, if your 13th chromosome and all has only one arm, the centromeric chromosome, the centromere is here and you just have the lower arm, Q arm. So if a part of that chromosome arm is moved to the 14th chromosome and settles on the as a P arm, people who can identify the 13th or 14th chromosome can easily judge that. So if not, FISH is a better technique to easily identify the translocations. This also helps explain poor ovarian response and recurrent implantation failure and pre-implantation genetic testing. Train embryo for during IVF. It can reduce implantation failure and miscarriage rates. Risk of chromosomally abnormal offspring can also be avoided. Advantages in reproductive medicine. Works on non-dividing cells with rapid results and high sensitivity for targeted chromosomes. Okay.
Can you go back? High sensitive. I'll try to give an example on this one, which is works on non-dividing cells. This is the case when our process was done. There were around 5 to 6 embryos that were formed.
Okay.
And obviously they had graded after like 5 days or something. Like one should become two to four, 4 to 8,
Cell division, yeah.
right? Whatever geometric proportion. Adila, if everything is normal,
Yeah.
divide works on non-dividing cells na, you are saying divide ஆகணுங்கிறதான objective. அது
Divide.
வந்து 2ல இருந்து 32 ஆகணும், right?
Correct, ma. அது என்ன சொல்றதுன்னா, before you even fuse the sperm and the ovum, அது non-dividing தானே. Only when you fuse it divides, right? Forms a zygote and forms a 2 cell, 4 cell, everything, embryo and all that. அதனால before it fuses, it's a non-dividing cell.
Okay.
Right?
Okay. Which goes back to which egg and which sperm to qualify.
Exactly, choose.
Okay. Understood. Okay.
That.
Rapid resultsனா இது எல்லாமே நம்ம வெளியில கொடுத்து அனுப்பணும். Depending upon the infrastructure, it can be done in operation theater or site lab, everything. Like டக்குன்னு.
Operation theater இல்ல, labsல, proper sterilized labs.
Sorry, அதுதான் operation theater பக்கத்துல lab இருந்தது, அதனால சொன்னேன். Meaning there's no need to send it outside, wait for results, etc. You do this fatafat. Okay.
ஆமா. ஆனா சென்னையில பார்த்தா I think Ramchandra has it and I think Mediscan has it. Mediscan genetic
we did our thing in Cloud9. I think that should have at the tertiary hospital na.
Okay.
Okay, fine. Okay.
Okay. Any other doubts? Guys, all good? Abhi?
Yeah, we have
Hi.
Shalini and Prasanna also joined. Just Shalini has given thumbs up. You can carry on.
Okay. Okay, now moving down to the next topic of the presentation, artificial reproductive technique or ART. It's a medical technique for people who are unable to conceive naturally. It's a scientific method to form an embryo inside or outside the human body. So egg plus sperm forms an embryo, and the types intrauterine insemination, IUI, IVF, in vitro fertilization, ICSI, and gift and zipped, gamete intrafallopian transfer or zygote intrafallopian transfer. So I'll explain each one to you guys. IUI, it's a okay, before that, the way you process a sample, it's through centrifugal method or swim up method or density gradient method. Centrifugal method is the most common processing method where you process the sample through adding buffer and centrifuging it and taking the required cells. Okay, the processing of the semen sample. You collect the sample, liquefy it at room temperature for half an hour. You assess the initial type of the semen sample, your volume, concentration, and motility. You dilute it with equal amount of washed medium or buffer. You centrifuge it at 2000 RPM, rotations per minute, RPM is, for 10 minutes, and the plasma remains as a supernatant. After discarding the supernatant, you add fresh medium, and then your second centrifugation starts, and that goes on for 5 minutes. Again, discard the supernatant and add 0.5 ml of your medium. You incubate it till further use at
Can
the
you
given
explain what
temperature.
is centrifugation here?
Centrifugation is
Centrifugation is like the same principle as your washing machine, where it spins and
Correct.
the
Understood.
heavier thing settles down,
settle down
and
and
the
your
lower
lighter ones
density one, it will be swimming on the top.
stop.
Understood. Understood. Okay.
IUI or intrauterine insemination. Samples collected from male. Processing of semen sample. And here the fertilization happens within the female body. So the sample is introduced within to the female uterus and fertilization occurs inside the body. So this is the protocol. Egg and the sperm fuse are in the uterus and fertilization happens in the tube. And then once the fertilization happens, they check after a given amount of time, it's checked through the usual protocol. IVF, here the same process, but the fertilization happens outside the body. You fuse the egg and the sperm outside the body using artificial conditions. And then once the embryo is formed, the embryo is transferred within to the female uterus. And then they give it a rest and then check through for pregnancy, and the normal process of pregnancy goes on if it's positive. But when the eggs are not proper, we do ovarian stimulation and all that to have proper egg size and proper quality of eggs to be obtained, right? When you want to uptake the eggs and all that, you have to have a proper quality of egg. So they do ovarian stimulation, and then the eggs are retrieved, sperms and the eggs are fused, prepared, and fertilization happens. And then the embryo is transferred, and then check for pregnancy. Success. Am I clear?
Yes. Just to again share my experience here, although this flow diagram is from 1 to 6 compared to the natural cycle, in each of the step, you have a decision or it is a probability game,
Make a
right?
decision, yeah.
That's the reason that you might hear your friends or colleagues or if at all people share with you the different cycles that they go through, the amount of agony or stress that they go through, the amount of frequency in which they would want to use these cycles, etc. Right?
True.
So, God and nature willing, both our cycles were successful in the very first attempt. Um, Again, it was 1 in a million. So we both our kids are one in a miracle in a way, 1 in a million. So, but again, each step has its own decision tree for
Own
you to
process.
decide something or the other, because in this sort of treatments, the hospital or the doctor who is consulting expects you to take such decisions upfront, or
Decisions.
either be clear on what you want to do. If something fails in 3, something fails in 4, are you
4,
ready
yeah,
for this? Are you ready for that? Etc. Etc. Right? So
exactly.
yeah, yeah.
When I was going through my pregnancies, one I miscarried, as well as when I was pregnant with my son. My checkup doctor was from Prashant Hospital. Okay, there when I go for checkup during the daytime, I see hell a lot of cases. The whole waiting hall will be filled with couples for just for this protocols, ICSI or IUI, IVF. When we hear their issues, it's really heartbreaking, because each person be like second time or third time with, or the eggs wouldn't have stimulated properly, they have to go again through the injection process and everything was like, I people who conceive naturally, I think that we have to literally thank God. But I didn't go through this big a hell, but I had my share of own hellish experience, because though I conceived naturally, I had to go through some injections and drips and everything. I have to go through to avoid all the previous issues, pain and all that. So post next after IVF, okay, ICSI, intracytoplasmic sperm injection. Your eggs are collected, your sperms are collected, but here the cytoplasm is sperm is injected in the cytoplasm of the egg. Okay, where the egg develops into an embryo once fertilized, and the embryo is transferred to the uterus. Yeah, your egg in, as usual, the process is here: stimulation of the ovary, egg retrieval, and sperm preparation. Once the sperm is injected into the cytoplasm of the egg, and once the fertilization happens, the embryo is transferred to the expectant mother, and then they follow the doctor's advice. Okay. Fifth, or zygote intrafallopian transfer, ovarian stimulation, the same process. The fertilization occurs in the lab. Your zygote or embryo is formed and transferred to the tube. The further development happens in the uterus. Here, the zygote is transferred, not the gamete.
Can you be a little specific there? What the difference I see is, you say the embryo was planted or transferred to the uterus there, but here it's fallopian tube. That is one difference.
Yeah.
But inside embryo, you are saying some gamete. What is that? What is
See, your egg and sperm are called gametes.
okay?
Okay.
Mhm.
Here,
And then you fuse them.
you fuse them, it forms a zygote.
Correct.
Okay, va. மற்ற process நம்ம straight uterus க்கு போடுறோம்.
Hmm.
இங்க but in natural process, once they both happen, the fallopian tube இருந்து தான் uterus க்கு போகும் நம்மளுக்கு.
ஆமா ஆமா ஏன்னா அது டிராவல் பண்ணிட்டு திருப்பி வந்து
பண்ணி அது
லேண்ட்
uterus
ஆகும்.
க்கு
லேண்ட்
land
ஆகும்.
ஆகும்.
ஆமா.
So that way, zygote, இப்போ they inject it as a zygote to the fallopian tube தான்.
Okay.
So it moves through the tube and embeds in the uterus.
Understood. அந்த எம்படே வந்து எக்ஸில் டைரக்டா பண்ணிடுறாங்க. This is letting it embed itself.
Embed itself, yeah.
Understood. Understood. Okay. Okay.
gamete intrafallopian transfer. Here, the egg and sperm are collected, placed directly in both egg and the sperm are placed directly in the fallopian tube, because some people won't have proper egg. So once they have to check for they give egg simulation for proper egg quantity and quality, and then they place both the proper egg quality and quality egg to the fallopian tube and the sperm as well, where the fertilization happens within the tube. The usual protocol, but here the process is selective.
Understood. Have you ordered this in terms in any particular order? I know it's complexity, but is ZIFT and GIFT more advanced than ICSI?
They are in the same type stream.
Hey, then why have I only heard of IUI and IVF? Like, why the other three are unheard of? Is it?
Andama, Abhi, IUI is very common in
That's
a
the
way.
primary.
Yeah.
That's the
Primary,
primary
right?
thing that we go for.
Which is almost என்ன சொல்றது கான்டெக்ஸ்டுவலா உனக்கு அனாலஜி சொல்லணும்னா there is no
Simpler one.
technical. It is almost like IUI is you are having the male material instead of a natural process. You are having the male material transferred inside the uterus and then leave it to nature, right? Where it goes swims
Exactly.
and then you go attack and then fuse. If it does not fuse, no. If it fuses, twins or
Yes.
whatever, right? That is IUI. IVF la you do this process outside in the lab, but
Yeah.
right? ICSI la வந்து the depending upon the complexity, for me that X chromosome microdeletion, sorry, X chromosome one band inversion ala the
Extra
quality
5
of
தானடா
huh?
5 தானே உனக்கு குரோமோசோம் இது இன்வர்ஷன்
fifth la P band ஏதோ P band
இன்வர்ஷன்
ஆமா inversion. அதனால the why IVF was not conducive for us, or at least from a commonsensical standpoint, is IVF la you are doing it in lab, not inside the body,
Yeah.
right? But you are still making the male material and female material approach each other and then fuse, which is again nature. You are not forcing it, or you are not selecting what sperm qualifies to what egg. Okay?
Yeah.
But in ICSI, in my case, the reason was the quality of sperms, or the probability of quality of sperms is what do you say is unpredictable. So they had to qualify what to select and then fuse it with the right egg in order to form the embryo. So it is a case of probability தான். In more of complexity oda the rarity of diseases தான். Disease சொல்ல மாட்டேன், rarity of the complexity தான். அந்த ஆர்டர்ல நான் என்ன கேட்கறேன்னா ஏன்னா இக்ஸியோட இந்த ஜிஃப்ட் கிஃப்ட்னு சொல்றது வந்து I have not heard of it because it is not been advertised, or it is not been talked about as an option.
See, most of the time it doesn't come as an option.
That's what I'm asking. Why? Is it because it's more expensive or labor intensive?
It is more expensive. It is more expensive.
Okay.
அதுதான். So ஆர்டர் பண்ணிட்டு வந்து in the range of you are saying gift is the most expensive, is it?
Yeah. I'm not sure. The price varies, I'm not sure. But technique wise, it is. They try. See, இப்போ IUI எல்லாம் நம்ம 2-3 தடவை ட்ரை பண்ணி அதுக்கப்புறம் தான் அது ஃபெயிலியர் ஆனா IVF போவோம். That's the way. If IVF doesn't work, then it's C.
ஆமா, எனக்கு கேஸ் எங்களோட காம்ப்ளெக்சிட்டினால the only solution was ICSI for us. That is
ICSI for you
separate
guys.
case, right?
ஆமா. So if there's no other go, fusing, இப்போ see gamete intrafallopian transfer வந்து where you use when your egg quality is not good, or the quantity of egg is not good, or see natural process conceive situation இருந்தா if you're not able to conceive, அப்ப ரெண்டுத்தையும் fuse பண்ணி ரெண்டுத்தையும் சேர்த்து they push it through the tube for you. So there you have no other go, you have to fuse ஆகும் அது.
Understood.
So அப்படி போகி the other zygote is formed, and this requires at least one proper functioning fallopian tube. ஏன்னா tubal block இல்லாம எதுவும் இல்லாம இருக்கணும், issues இல்லாம இருக்கணும். abnormal tubes இருந்ததுன்னா this process is tough.
Understood.
Here I even have to add one thing. Me being born was literally a miracle here, because my mother had both the tubes blocked. but there was very minor spill in one tube, and the doctor said there were very mild chances of her conceiving. By God's grace, she conceived, and that too after 14 years of her married life. And I was born with, and with that too, with God's grace, no genetic issues, since both paternal and maternal age are beyond 30s. My mom was 36 when I was born, and my dad was 43. So there are a whole lot of chances of genetic abnormalities that could have come.
What is a common reason that they say statistically greater than 30, greater than 40? The more we age, the quality of the materials go down, huh? Or is that
See, there are chances of chromosomal damage. See, your lifestyle changes.
so okay. Chromosomal damage as we live. It is not that what we got when we were born.
What we got, no. அது
For example,
இருக்கிறது
if in case I'm an I have say once a week alcohol,
yeah,
that will go and damage in some sense or lens.
exactly. And at
Okay.
the same time, when people are more exposed to radiations and
Mhm.
all that also damages your chromosome, which is
So,
susceptible to breakage and more damage.
hmm. Okay. Before you're exposed to what age takes you in life, better
Yeah,
put the point here. Okay.
it's better.
Okay.
And those years, it was 30s and 40s. But this generation, our generation, it's literally kind of late 20s to starting of the 30s. When
Actually, it has no. Actually, it has increased. My doctor said, because in my mom's years when she studied, it was above 30. Now it is advanced gestational age or something, and the
gestational age.
gestational age. But now it is beyond 35. It is increasing as generations go by. It is increasing because more and more mothers get pregnant more later in their lives.
Probably, Abhi, they can freeze their eggs.
See, I went to a doctor in Houston and I asked, can I freeze my eggs when I was 32? And
Yeah.
she was like, why the hell do you want that? She said, poor apokla pata, I don't think I'll get married. I just want to freeze it and live a happy life. Yeah, I literally told her that. I just want to freeze it and live a happy life. And whenever I want to, I can unfreeze it. And then she was like, just go enjoy your life. Don't think about all these things. Just go. She was a Patel doctor. She was a Guju doctor. And then she just said, I'll give you some sleep medicines and that's what you need. Just go have some night sleep. So, and then I got married at 34 or 35, then I, had the baby at 36. So it really doesn't, I think it depends on person to person.
Yeah, it's individual choice. I know people
It's
who are
individual choice,
for
and
whatever it can be their career choice, it can be related to their some other commitment, etc. People at 25 to 30 are still visiting doctors, convincing them, or if the protocol allows, why not? Question கேக்குறாங்க இப்போலாம். Right?
Yeah.
Right?
true.
So, yeah.
But not all the people have who are 30 plus or 40 plus have kids with defect. And at the same time, not all the people who with 25 and 30 age limit don't have perfect kids
Yeah.
without defect and all.
Yeah.
Because when I was in my college and I had pediatric rotations, I have seen small babies with cleft lip and cleft palates.
Yeah. Yeah.
What with what? Small
Cleft
babies
lip,
with
cleft
what?
lip.
Cleft lip, that's your broken lip. Your
Okay.
lip
Okay.
and the palate both would be damaged.
Okay.
Yeah. But another, it's a minor plastic surgery. They will correct it now.
At sometimes it might have be even genetic.
Yeah, it is genetic.
Yeah. அது correct பண்ணலாம். But even with that issue also is a thing, right? அது கூட damage ஆகுது இல்ல chromosome damage ஆகுறதால வருது இதெல்லாம்.
Yeah.
Understood.
So not all the people are susceptible, not all the people are risk free. That's what you can say.
Got it.
Yeah.
So advantages: helps infertile couple. You can overcome male and female infertility problems. Helps prevent genetic disorders. Allows delayed parenthood. As Abhi, as you said, delayed parenthood is also added is an advantage here.
Yeah.
option for single parents or same-sex couples.
Yeah.
Who want to be single parents, they can also have this as an advantage, and same-sex couples are also at advantage for this. High success rate with advanced techniques. You have a very high success rate with advanced technique. As your techniques advance, there's a high success for this. Yeah, that I'm done with this.
Understood. I just wanted to share something in this what option for single parent example. Again, is what we saw witness in the hospital. Again, a very
Yeah,
extreme case, but just to know what the world is, what people are facing, right?
true.
I think they did whatever cycle, it can be IUI or something else or XE like us, etc. But whenever that embryo is formed, you get a form, right? Maybe I think in the first cycle, I think we were starting with 8, and then towards the grading, 5th day, 6th day, because of the first cycle, they implanted 2. God and nature willing, 1 formed, hence my daughter. Okay. Second, we were particular on not don't put 2, or rather we don't prefer. If in case it forms a twin, already one முடியல. So we told only one. We told only one. If it forms, it forms. If it does not, me and Sridevi have decided that we will not repeat உடனே will take 2 years, 3 years if required, or we will not even proceed. இப்படிங்கும்போது அந்த let's assume that 8ல only 5 has qualified at the end of the 5th day before you transplant or before you implant, sorry. um, so 2 were put, 3 were remaining. So this 3 is actually frozen, and then
Yeah,
I
they
think
can
it's
do that.
technically valid for 5 years or something. You
True,
can
true. They can do
thaw
that.
it, defreeze it, and then use it for your next cycles. அப்போ
Exactly.
formula there are options, right? Which is you have to once you are successful on the cycle and then you conceive, depending upon your own comfort level, you can say use it for research purpose, the remaining embryo, right? Or you can discard it or whatever. அதுல I think that is, if in case you need to store, ஏதோ நாமினி அந்த மாதிரி ஏதோ போடணும்னு நினைக்கிறேன். Okay, I think we were not having a problem, but just to show paint a picture of extreme case, I think there was one couple who went through some cycle, and then they had their first kid, uh, but they had embryos frozen.
Okay.
Okay. Uh, 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 very, very unfortunately, the husband passed away.
Okay.
Okay. The lady came back and then asked for this. I need to start my second cycle.
Second
And then
cycle.
for some reason, I think he did not put that I give my consent to my wife to, etc., etc. If in case there is extreme situation, and then by protocol they were not supposed to give, right? They were not supposed to, and I think that the lady had to live with that one child, not necessarily try for the second. So smaller nuances, again, beyond whatever we studied or learned today, process wise, again, it's almost like you what to say, you create a 2 crore corpus in a mutual fund, but don't put nominee, you're gone, nobody gets the money. அந்த மாதிரி தான், right? So
See, and at times this is also used where specific பையன் தான் வேணும் இல்ல பொண்ணு தான் வேணும்னு சொல்றாங்க இல்லையா, அந்த டைம்ல கூட they can, because the sperm finalizes if it's a girl or a boy. X இருக்கிற sperma வந்தா girl, இல்லன்னா it's a boy. If you get a chromosome with bi, if you get a sperm with bi chromosome, it's a boy. So
ஆமா.
that way also some people who are very particular with பையன் தான் வேணும் பொண்ணு தான் வேணும்ன்றதுல, that way also they can use this. But
they nobody discloses that, no, there's
that's true. They don't disclose it here, but at some places apart from India where they don't kill the female
once
child
the
and all
embryo
that,
is formed, before
exactly,
you can choose, is it? Okay, understood.
they can do that.
Understood. Okay. Oh, yeah, it depends on the society and
Society and the rules
okay.
and ethics.
Understood. Yes, obviously. Super.
Here in India, disclosing the sex of the fetus is crime. But
Yeah.
whereas in abroad, you actually disclose the sex of the fetus.
Yeah.
Am I right, Abhi?
Yes, of course. They had
Yeah,
gender revealing parties.
gender revealing parties and all with all the blues and pinks done.
Yes.
Yeah.
So
Like
depending on the rules.
whenever I face there are so wide இப்படி இருந்தா அப்படி இருக்கும், wide இப்படி இருந்தா இப்படி இருக்கும். My ayah is telling
I
something.
had that. I
My
had that.
my in-laws are telling something. My some party in village is telling something. Sridevi, whoever tells that, I tell only one thing, right? 10 ஆப்ஷன் இருந்தா கூட பிரச்சனை நீங்க சொல்றதுல ஒரு லாஜிக் இருக்கு.
No,
இருக்கிறது 2 ஆப்ஷன். ஒண்ணு இது இல்லாட்டி அது. இதுல
அது
என்னங்க உன்னோட சயின்டிபிக் லாஜிக் பண்றீங்க அப்படின்னு சொல்லிட்டு. ஸோ
no, I had that because when I was pregnant, both the times it was boys. Okay, the one I miscarried twins were also boys, and the last pregnancy with my son was also okay. But I didn't know till the end. But my stomach was not going this way. I mean, not wide this way, but this way. இதுக்கெல்லாம் ஒரு இது இருந்தது. இப்படி போனா பையன், இப்படி போனா பொண்ணுன்றதெல்லாம். But at times it goes wrong.
yeah, there's no, it's all
And like
conversation.
whatever it is, I don't mind. I just want the child to be healthy.
Exactly. Super. Okay, I think we had a fantastic session. Some, I think some of us might have experienced little technical concepts. It's not new for me, but depends on how people's experience and exposure are, and real life situations also. Thank you so much, Jeevitha, for doing
Thank you.
this. I'm just stopping recording.
Did I stammer so much or struggle so much?
No, I think you are super
Yeah,
fluent.
it's been 13 long years since I touched all this topic.
Okay.
Okay, I am not working anywhere, but I just wanted to share so that people can understand this and they can go through the whatever process they have to go through if they have any issues with pregnancy and all that without any fear or trouble.
Yeah, it's almost equivalent to you knowing somebody who has gone through. At the same time, I think with over time, it's just a matter of time that our friends or relatives, etc., right? And it's in some lens, all this was or is actually is taboo only, not necessarily, right?
Yeah.
We are like 6 or 7 people right now. We are openly talking and sharing stuff. Not the world is not like that, right? And life being not necessarily fair to everybody every time. Who knows what you need to know to a situation, right? For anybody, for that matter, not only for you. So
Yeah.
அந்த கான்செப்ட்ல தான் இந்த லேர்னிங் செஷன்ஸ், ரைட்? யூ நோ நெவர் நோ வேர் வாட் வில் கெட் யூஸ்ட் இன் டர்ம்ஸ் ஆஃப்.
ஏன்னா எல்லாமே தெரிஞ்சு when I was pregnant இதுவா அதுவா I had all those thought process going in mind. At times I used to think எதுவுமே தெரியாம இருக்கிறது இன்னும் நிம்மதியா இருக்கும்ன்றது.
innocence is bliss.
Innocence is much better in some point.
Yeah. Me being me, I think just before our first cycle, before because there are so many decision trees that we had to do, and it pushed me. I'm very curious person, but it pushed me to think extreme scenarios. It was an opportunity for me and wife to converse scenarios which we can't even imagine, right? What if this happens? What if this happens? What if that happens? Are you okay? What
It's
is your
actually
standpoint,
stressful for the
etc.,
couple.
right? அதெல்லாம் போட்டு நான் பிபிடி என்னமோ டிசென்ட்ரிலாம் போட்டேன், ரைட்? அந்த டிசென்ட்ரியை நான் வந்து டாக்டர்ட்டு காமிக்கல. தட் இஸ் ஒன்லி திங் தட் ஐ ஹவ் நாட் டன், ரைட்? பட் given all the conversations, the doctor was like, in my 25 years of experience, I have not even seen anybody who asked so many questions. But my answer or my thought process was simple. First point, this is not a compulsion. This is an option that I'm taking.
Yeah.
Right? If in case it's cancer or something for some reason, I had to live with. There's no other option, right? So I need to know, or me and Sridevi need to know what are the nuances. You can easily ask in the scenario என்ன next sitting 2 weeksல நீங்க டிசைட் பண்ணி சொல்லுங்க, நீங்க ஈஸியா சொல்லிட்டு போயிடலாம். But there's not an easy
No,
answer.
they
Right?
say the couple have to be stress free. Anna,
Yeah.
this is the most stressful situation the couples face.
Yeah.
The doctors say that easily. எனக்கும் என் கேஸ்ல எனக்கு நான் ஜெனடிக்ஸ் பேஸ், எங்க அப்பா டாக்டர். both of us were like discussing இதுதான், அப்பா இது இருக்குமா அது இருக்குமா இது இருக்குமா அது இருக்குமா. All that was what is going on.
Yeah. Super. I'll stop recording. Thank you so much, Jeevitha.
Thank you.